From Hiranandani College of Pharmacy, I am going to discuss about antifungals today. I am going to deal with the general introduction, their classification, their mechanism, structures, and ACR. So, how to define the antifungal agents? The antifungal agents are simply defined as the agents which are used against fungal infections. They are also called as antimycotic medications. Now, there is a basic structural difference between the fungi and the human cells, and the difference is cell membrane is made up of the lipid bilayer. We all know that. In the case of humans, this lipid bilayer contains cholesterol as a main constituent, whereas in the case of fungi, this lipid bilayer contains ergosterol as their main constituent. And because with this difference, we come across with various fungal infections, which include the severe skin, eye, or ear infections, or the infections of other body parts like athlete's foot, ringworm, candidiasis, and various serious systemic infections. Therefore, the primary mechanism of action of antifungals is the inhibition of ergosterol biosynthesis. So, how to classify the antifungal agents? They are classified as fatty acids and related compounds, phenols and related agents, antifungal antimetabolites, antifungal antibiotics, allylamines and related compounds, and azole antifungals. So now just begin with the fatty acids and related compounds. On our skin, we have the layer of sebum. So this naturally present sebum on the human skin is acidic in nature, and the function of that is a natural antifungal agent. So chemically we have designed the fatty acids which are very similar in the structure to sebum. So the idea is we have designed an agent which are similar to the sebum in structure, therefore they will augment the function of sebum, or they will help the sebum and the fungi can be eradicated. So when these fatty acid and related agents are because of their chemical similarity to sebum, when applied on affected areas, they will augment the sebum function and fungi can be eradicated. The examples of these agents are the sodium caprylyl and zinc and decanylate. The next agents are the phenols and the related compounds. Now, majority of these compounds have a topical activity or can be used for topical infections. It has been considered that they are interfering with the fungal cell membrane integrity. They are responsible to decrease the surface tension of the fungal cell membrane, and because of which the fungal cell become weak and they die. So the agents are haloprogen and the siloquinol, whereas siloquinol is very famous for the treatment in athlete's foot and the vaginal infections. The next important agents are the antifungal antimetabolites. Now they are called as antimetabolites because they have a structural similarity to the very important cell constituent of the fungi. And they have a structural similarity to these agents, and because of these structural similarities, the fungi takes these synthetically prepared agents or the drugs as a substrate in the natural processes. So just let us understand this. So they are also called as nucleoside antifungals. They have structural similarity to the pyrimidine basis of the fungi. So example is a 5-fluorocytosine. The structure is shown here. The structure is very similar to the cytosine base. So this 5-fluorocytosine enters the fungal cell by active transport mechanism and gets deaminated by the cytosine deaminase, and the resultant is the formation of 5-fluorouracil. Again the structure is shown in the scheme. So this 5-fluorouracil will undergo the phosphorylation to form 5-fluorouracil deoxymonophosphate, and this 5-FUDUMP have a structural similarity to uracil. So this uracil in natural way or a natural process gets converted to thymidine, and another important pyrimidine base which is a major constituent of the RNA. So this uracil to thymidine conversion is by thymidine synthase. So now what happens is our 5-FUDUMP have a structural similarity to uracil. So the thymidine synthase takes 5-FUDUMP or takes 5-fluorouracil monophosphate at the place of uracil, and thymidine synthesis is inhibited. So the resultant is RNA synthesis is stopped and the fungal cell will die. So this is how our 5-fluorocytosine is helpful as an antifungal antimetabolite. The next important agents are the antifungal antibiotics. There are two types of antibiotics, pauline antibiotics and the griseofulvin. Now just look into the paulines. Now as the name suggests, polyins means they contain many double bonds. Poly stands for many and in stands for double bonds. So they contain many double bonds in their structure, and therefore they are named as paulines. Now paulines are macrocyclic lactones which contain many conjugated double bonds attached to deoxyaminohexose sugar like mycosamine through glycosidic linkage. there are either 26 membered polyenes which are named as natriumycins, and there are 38 membered polyenes which are called as anti-amphotericin B or nystatin. Now their structure contains two important parts. The first part, at one side, they contain the hydrophilic hydroxy groups which are giving the weakly acidic character to the compound, whereas they also have the lipophilic conjugated double bond at the another side. So what polyenes do? Due to barrel-like lipophilic part of the structure capped with sugar, they penetrate in the fungal cell membrane and orient all polar hydroxy groups towards each other. This acts as a false membrane component and binds closely with ergosterol and disrupts the fungal cell membrane, causing all the cell constituents to leak out of the cell. So these are the structures of the amphotericin B and the nystatin. Again, the biological sources of amphotericin B are also mentioned here. Amphotericin B is obtained from the Streptomyces nodosus, whereas nystatin is obtained from the Streptomyces norisii. Now, another important antibiotic in the fungals or the antifungals is griseofulvin. Now the griseofulvin is a spiro compound. We can see here the junctional carbon between the 5-membered ring and the 6-membered ring. That carbon is called as a spiro carbon. So griseofulvin is a mitotic spindle poison. It affects the mitosis process or a cell division process in a metaphase. It causes rapid and reversible dissolution of the mitotic spindle apparatus by binding to tubulin. The biological source is Penicillium griseofulva. The another important agents in the case of antifungals are the allylamines and the irritate compounds. Now, as we know that fungal cell is made up of ergosterol, so all the agents now hereafter will affect the ergosterol biosynthesis. So allylamines are one of them. Allylamines interfere with ergosterol biosynthesis in an early state, namely epoxidation of squalene. So they are squalene epoxidase inhibitor, we can say. So inhibition of the squalene epoxidase stops ergosterol biosynthesis, which leads to squalene accumulation in the fungal cell. Resultant is destabilization of the cell membrane. So these allylamines and the related compounds are fungicidal against the dermatophyte infections and the fungistatic against the filamentous fungi infections. So these are the agents: naftifine, terbinafine, butanafine, and a tolnaftate. Now in this case, naftifine and terbinafine have that typical 3 carbon containing double bond chain which is allyl, and they also have the amine. Therefore these agents are called as allylamines. In the case of butanafine, we can say there is an amine, and next to amine there is a 1 carbon attached to benzene ring. Now if you can clearly look into the benzene ring and the carbon which is attached to amino group, we can clearly picture out the allyl part of the structure. So butanamine is also an allylamine compound, and the other example of these azole antifungals. So the azole antifungals are named as lanesterol 14-alpha demethylase inhibitors, which affect the ergosterol biosynthesis. So lanesterol 14-alpha demethylase is one of the cytochrome P450 enzyme required in the biosynthesis of lanesterol and subsequently to ergosterol in the fungus. So just look into this scheme where the squalene subsequently get converted to the lanesterol, the intermediate formation of squalene epoxide through squalene epoxidase. So now what happens is the steroidal molecule contains the 5-membered ring, and that 5-membered ring in the steroidal molecule will interact with the active site of the 14-alpha demethylase, which is shown in the diagram. The active site of the 14-alpha demethylase contains heme as the cofactor, and this heme cofactor interacts with the methyl group of the 5-membered ring, 5-membered ring of the steroid. So now, however, azole antifungals helps us. So azole antifungals contain the nitrogen which is containing lone pair of electron. So this lone pair electron in azole antifungals bind to heme active site and prevents the oxidative demethylation of a lanesterol, or rather prevent the formation of lanesterol. Again we refer this diagram where the structure of the azole antifungals has been shown, and it has been also shown that nitrogen of the lone pair in azole antifungals form a bond with the 14-alpha demethylase active site which contains heme, exactly in a similar manner as 14-alpha demethylase in natural sense form a bond with a steroidal biomolecule. So ultimately the result of this is the inhibition of lanesterol biosynthesis by inhibiting the 14-alpha demethylase enzyme. So when demethylation is inhibited, there is an accumulation of 14-alpha sterol. This 14-alpha sterol accumulation causes the membrane destabilization and initiate the chitin synthesis. This initiation of a chitin synthesis further degrades the fungal cell membrane. So this is the simple mechanism of the azole antifungals. Now just look into the structure and ACR of the azole antifungals. The general structure of the azole antifungals has been shown here. Now generally the azole antifungals contain either imidazole or either 1,2,4-triazole as their basic moiety. These agents contain either 2 or 3 aromatic rings and the agents when contain 2 or 3 aromatic rings which are heterocyclic aromatic rings are highly potent. At least one of the heterocyclic rings should contain the halogen, and amongst the halogen the fluorine increases the activity. It has been also seen that when the halogen is present at the second position or the second and fourth position in the case of di-substituted compounds on the aromatic ring, the compounds are highly active. Introduction of halogen at any other site, any other than the second and fourth, responsible for inactive compounds. So these are the azole antifungals which are very good or very potent. Out of this, clotrimazole, miconazole, flutrimazole are the also our cetraconazole are the flammable ring compounds, whereas ketoconazole, itraconazole are the 1,2,4-triazole containing compounds. Thank you.